This is Dr. Divya. Today's topic is biotechnology and its applications. So, first of all, what is biotechnology? The term biotechnology was coined in 1970 by engineer Carl Erke to describe a process for large-scale production of pigs. Biotechnology can be defined as an application of technology using the living organism to obtain useful products. So, what is biotechnology? Biotechnology is a multidisciplinary in nature involving input from engineering, computer science, cell and molecular biology, microbiology, genetics, physiology, biochemistry, immunology, virology, recombinant DNA technology, genetic manipulation of bacteria, viruses, fungi, plants, and animals, often for the development of specific products, biostates, enzymology. So, biotechnology is the application of biological organism, system or process to manufacturing and service industries. It is an integrated use of biochemistry, microbiology, and engineering sciences in order to achieve technological applications. It is the controlled use of biological agents such as microbes and cellular components. So, development of biotechnology, ancient biotechnology, early history related to food and shelter include domestication, classical biotechnology, fermented food production and medicine, modern biotechnology manipulates genetic information is an organism, genetic engineering. So, basically, DNA, the molecular of life. This is a chromosome, gene, cell, so, genetic engineering. So, it is an in vitro DNA technology used to isolate genes from an organism, manipulate them in laboratory as per desire, and insert them into other cell or system for specific character. So, it is called gene cloning. Recombinant DNA technology is one of the recent advance in biotechnology, which was developed by two scientists, Boyer and Cohen. So, Cohen, who received the Nobel Prize in Medicine for his work on discovery of growth factor, and Boyer, who constructed the first recombinant DNA using bacterial DNA and plasmid. So, RDT, the recombinant DNA technology, works by taking DNA from two different sources and combining that DNA into a single molecule. That alone, however, will not do much. So RDT only becomes useful when the artificially created DNA is reproduced. This is known as DNA cloning. So in the early 1970s, technologies for the laboratory manipulation of nucleic acid emerged. So in return, these technology led to the construction of DNA molecule composed of nucleotide sequence taken from different sources. So the product of these innovations like RDT open exciting new avenue. So concept of RDT, recombinant DNA is a molecule that combines DNA from two sources, known as gene cloning, creates a new combination of genetic material. Human genome for insulin was placed in bacteria. The bacteria are recombinant organism and produce insulin in large quantity for diabetic. Genetically engineered drug in 1986. Genetically modified organisms are possible because of the universal nature of the genetic code. So RDT begins with the isolation of gene of interest in target gene. So the target gene is then inserted into the plasmid uh, vector to form replicon. And replicon is then introduced into host cell to cloned and either express the protein or not. The cloned replicon is referred as a recombinant DNA and the procedure is called recombinant DNA technology. So cloning is basically necessary to produce numerous copies of the DNA since the initially supply is inadequate to insert into host cell. So some other term are also used in common use to describe genetic engineering like gene manipulation, recombinant DNA technology, gene cloning, you can say molecular cloning also, genetic modification. So six steps of recombinant DNA. First one is isolating, second one cutting, third one joining like ligation, and fourth one transforming, fifth cloning, and sixth selecting. So DNA, basically DNA cloning is a plasmid vector permits amplification of a DNA fragment gene for that inserted in antibiotic resistance, and this is the foreign DNA region of interest. Basically that plasmid have a sticky end, and they foreign DNA and that plasmid hybridization with the help of a DNA ligase, they make a recombinant DNA, and that DNA inserted into host cell. After that cloning process is started. So this is the process of cloning into a plasmid. So this is a process. Plasmid vector permits amplification of a DNA fragment. So this is the process. Bacterial chromosome, the plasmid isolated, and here cell containing gene of interest, DNA isolated, then gene inserted into plasmid, and plasmid put into bacterial cell. So that make a recombinant bacterium, and after that cell multiplies with the gene of interest, and they make a lots of copies of gene, and then protein used to make a snow foam, like at a higher temperature, protein used to dissolve blood clots in heart attack therapy, and gene also used for alter bacteria for cleaning up toxic waste, and gene also waste used for pest resistance inserted into plants. So these are the applications of recombinant DNA technology: analysis of gene structure and expression, pharmaceutical products like drugs, like for example human insulin, and other one vaccines, recombinant vaccines. First one, genetically modified organism, GMO. First, transgenic plants. Second, transgenic animals. So analysis of gene structure and expression using specialized recombinant DNA technique, researchers have determined vast amount of DNA sequence, including the entire genomic sequence of human and many key experimental organisms. The enormous volume of data, which is growing at a rapid pace, has been stored. So first one, the gene bank at the National Institute of Health, Bethesda, Maryland, and the EMBL sequence database at the European Molecular Biology Laboratory in Heidelberg, Germany. So pharmaceutical products, some pharmaceutical applications of DNA technology, like first large scale production of human hormone and other protein with therapeutic uses, and production of safer vaccines. A number of therapeutic genes like insulin, interleukins, interferon, growth hormone, uh, erythropoietin, and coagulating factor like coagulating factor 8 are now produced commercially from cloned gene. Background of recombinant insulin: first, diabetes increased glucose concentrations when 80 milligram. Serious complications cause nephropathy, early years insulin isolated and purified from pigs and cow, and like leads problem like allergy in some people, sacrifice of animal. Attempt to make recombinant insulin started in 1970. So technique involves inserting human insulin gene in promoter of lac operon of the plasmid of E. coli bacteria. So first, normal glucose stimulated insulin secretions, that the process glucose, the glycolysis, in the process of glycolysis they make a pyruvate. After that the process happen. And second one, impaired glucose stimulated insulin secretion in VHL knockout. This is the structure: the promoter first like Z gene and promoter second like Z gene. This is the insulin gene A subunit and B subunits. So that transform into E. coli host cell. After that, fusion protein accumulates in cell. So pharmaceutical companies already are producing molecules made by recombinant DNA to treat human disease. So recombinant bacteria is used to production of human growth hormone and human insulin. So some protein produce of recombinant DNA technology, human insulin. This is a got from E. coli. This is uses of treatment for diabetes, human growth hormone, treatment for growth defects, epidermal growth factor, treatment for burns and ulcer, interleukin 2, possible treatment for cancer, bovine growth hormone, improving weight gain in cattle, cellulase, breaking down cellulose for animal feeds, and Taxol, this is the use for